So for the rest of the lecture, I want to talk about a particular study, neo-sex chromosome in Drosophila. So when I say neo-sex chromosome, that means the chromosome, the sex chromosome, is new in this lineage compared to the related species. The reason for this type of study is we can never go back to evolution to really look at the different status of the sex chromosome. The comparative study allows us, for example, from the human XY analysis, we try to hypothesize the possible evolutionary phases of this pair of sex chromosomes. And by comparing the other lineages like monotrans and the marsupial, it seems like just like a double confirm our hypothesis, and so that's like a new observation. And those lineages resemble some of the ancestral states of this XY system. Study neo-sex chromosome in Drosophila particularly, it allows us to try to look at the sex chromosome evolution in action. That means we knew this neo-sex chromosome just evolved during, like I say, several million years. And we try to understand the differences between this newly incurred region compared to the autosomal status between close related species. Showing in this slide is the difference between, say, Drosophila miranda and Pseudoobscura. And you can see the blue part indicate a fusion of the autosome to the ancestral sex chromosome. The next, and this has happened several times in the Drosophila lineage. The miranda is the one well studied in Drosophila, and also there's Virulus, which has a very ancient neo-sex chromosome, and also Drosophila abamicans, which the neo-sex chromosome evolved very, very recently. So that's just a different time scale. And in Drosophila americana, that's in the middle, the neo-sex chromosome is even polymorphic within the species. So the fusion between the ancestral sex chromosome and the autosome is polymorphic. It's not fixed in this species. So all these lineages indicate different status or different phases of the chromosome evolution. And this, we can use those materials to understand what type of mechanism or what type of mutation took place in evolution at a different time scale. So the blue part tries to illustrate the time scale on this phylogenetic tree to indicate if we study all these neo-sex chromosomes, we probably can understand how the sex chromosome evolved at the molecular level. Okay. So this slide just illustrates what is the neo-sex chromosome and the evolution of the neo-sex chromosome in Drosophila. So A showing the state of like a pair of XY chromosome and the autosome. So that's like the situation as in Drosophila melanogaster. Then in some lineage, it will be one pair of autosome, actually only one of the autosome fused with either X chromosome or Y chromosome, showing in the B status. And so you can see there's a neo-Y chromosome. Actually, neo-Y is the fusion between an autosome and the ancestral Y chromosome. And what's interesting by genomic study, people already discovered actually this Y chromosome now, this is the Y chromosome now is entirely the neo-Y chromosome. This is illustrated by the color. So you can see the Y chromosome in the B status. Now the entire Y chromosome is from the neo-Y region. That means has more homology to the ancestral autosomes than its original Y chromosome. And the original Y chromosome is highly reduced in this species. So you can further down to the C, you can see that the degenerate neo-Y chromosome, so the Y chromosome become reduced in size. But now in this species, now we recognize X and Y. Actually, the Y chromosome is completely different compared to the like a more ancestral state that's in Drosophila melanogaster. What we learned from Drosophila miranda, which is a well-studied neo-Y chromosome, and we observe, means like the scientists work on the species, observe there's accumulation of transposable element, and this was discovered before any sequencing involved in this species. So they look at the transposable element and they use classical hybridization technique to see the accumulation of transposable elements on this neo-Y chromosome. So there's an autosomal region fused with the Y chromosome, and now they look at this neo-Y chromosome portion, and there's a lot of transposable element insertions compared to the homologous chromosome. So the homologous now we call neo-X chromosome. So that's like the very first evidence of the degeneration of the neo-Y chromosome in Drosophila miranda. The second observation is by sequence several loci on the neo-Y chromosome and compare the evolution rate between neo-X and neo-Y. They observed the fast protein evolution rate, and that was discovered by higher DN-DS ratio, which should be familiar with you. And this was a study done by Betrol in 2003. And just by picking up several loci on the neo-Y portion and do evolutionary analysis on those genes, and she discovered, and there's a higher DN-DS ratio, means there's more accelerated protein evolution for those loci. And further, she also observed codon usage bias. There's lower codon usage bias on the neo-Y chromosome. And they found in several genic regions there are frame shift mutations, so that's a sign of a degeneration of the genes, and also like a result in premature stop codon. And for other functional loci, she also observed there's a reduced expression of the neo-Y alleles. So given the studies in Drosophila miranda for their neo-sex chromosome, it seems contain all the features we observed in a human Y chromosome. So there's lower gene numbers, and so the mutations create frame shift and stop codon, so that degenerate the loci, so the loci become non-functional on the Y chromosome compared to the X chromosome. And also there's a process tool non-functionalization, also in agreement with non-functionalization, is the reduced expression of the neo-Y alleles compared to the neo-X alleles. So this just show to allow us to understand the early evolution of the sex chromosome. Of course, this is a neo-sex chromosome, but we think it's probably very similar mechanism also happened during the evolution of the human Y chromosome. One thing I forgot to mention is the fusion of the autosome to the sex chromosome in Drosophila, particularly the Drosophila Y chromosome. The recombination stopped immediately between the neo-Y portion to the neo-X portion. The reason is there's no, in most Drosophila lineage, there's no recombination in males. So once the chromosome fuse with Y chromosome, they only evolve in the male, and there's no recombination between the neo-X and neo-Y. So the recombination suppression occurred right after the fusion event. And this slide is to illustrate the dosage compensation. So after the fusion of the autosomal region to the sex chromosome, and one thing the genome has to deal with is the dosage compensation as the original X and Y system. So the current hypothesis is also the evolving of the target affinity sites, and also that spreads through. So it's similar to what happened in the males, and so there's a dosage compensation complex compared to the females. And the remaining question is, in the early stage of the neo-sex chromosome, is this model also occurred on those Y chromosomes or neo-Y chromosomes? We don't know, because this model is more to the established XY system, how the dosage compensation is controlled. But if an autosomal region just includes as part of the sex chromosome, how the dosage, how the gene dosage was regulated is still an open question. I think more study is needed in this area to understand how the dosage compensation can spread through the entire chromosome. But a lot of the scientists sort of hypothesize this has more to do with the original or the existing dosage compensation system in the organism, and also the chromatin status. So chromatin status also be part of the gene regulation system. So this is something remain to be further investigated.